Welcome to Inside the Studio. I'm Darren Woodside, the Vice President for Research here at the Texas Heart Institute, and I'm with Dr. John Elrod. Dr. Elrod is an associate professor at the Katz School of Medicine from Temple University. He's also the director of the Cardiovascular Research Center at Temple. Welcome, Dr. Elrod, and thank you for a great Grand Rounds presentation today for our series that we have every week. Based on your work, that's really been fundamental in understanding the biology of these little organelles inside cells called mitochondria, and the impact that they can have on things like injury to the heart and chronic heart failure patients. For our audience, I was wondering if you could just kind of describe in very general terms what mitochondria are.
Sure.
And how they influence the outcomes in patients that might have an acute myocardial infarction or patients with heart failure.
Yeah, so thanks for having me. The setup and the experience at Texas Heart has been unbelievable so far. The lab focuses on mitochondria, a small organelle within the cell. What's unique about it is it has two membranes, and everybody probably learned in biology class that it is the powerhouse of the cell.
Right.
So most of ATP,
the energy provider.
Exactly. most of ATP generation, the energetic currency, if you will, is produced by the mitochondria. In addition to that, and the reason our lab is interested in studying mitochondria is it's a primary signal also for cell death and also a number of other newer cellular processes that we're starting to understand. And what I talked about today was how the mitochondria can also participate in having metabolites regulate gene program activation or changes in gene expression, meaning the underlying changes for protein changes in a cell and what a cell's trying to do. And so we really look at it as a hub, a signaling hub for many different processes: energy, cell death, and also communication to the rest of the cell.
So I guess if you consider ATP the energy monetary unit of the cell, the mitochondria is kind of like the central bank, right?
It is kind of the central bank, yes.
And it's a different kind of way of examining how cells are influenced by things inside the cell outside of just pure protein expression and gene expression, because it seems like the mitochondria provide a lot of things, metabolites, that can actually affect downstream gene regulation that can cause cells to differentiate and turn into different types of cells that might have beneficial or, in this case, bad effects on heart function.
Yeah, that's one of the things we're interested in studying. So the field for a long time has understood this dichotomy of energy production, versus cell death. And in these chronic disease states like we're talking about, such as when you have a heart attack or heart failure, really you have an imbalance in both of those things. You have an increase in cell death typically and an energy deficit. And so really trying to unravel, the pieces of that and how that actually plays into how a cardiomyocyte, the functional unit of the heart actually is impacted is something we're really interested in. And then ultimately how to therapeutically try to target that. So as you mentioned today, the talk was about fibrosis, so how mitochondria inside fibroblast, the cell that deposits what we call extracellular matrix. And I guess for the lay audience, the general idea there is the fibroblast is responsive to wounding, and so it's a wound healing response. And if you think about a cut on your skin, you start to get deposition, of the matrix, or you get a change in the deposition of the matrix, and then you start to get contraction of that wound to close. Same thing happens in the heart, but in the context of heart failure, that becomes maladaptive in chronic states. And so we're trying to understand ways to block that or revert that change.
So when you're thinking about these different ways, you come up with a strategy to maybe target a specific pathway, or in this case, a specific generation of metabolite. How do you take some of the basic research that you are doing now and translate that into something that could be of therapeutic value to a patient?
It's a great question. So our lab first leans heavily on animal models. So to verify a given target, we try to genetically manipulate that animal in a cell-specific way to see if in fact we can block that pathway, and if that then is in fact therapeutically profitable. In other words, does the animal actually do better in the context of heart failure? In this case, I'm talking about mice. As far as really moving towards therapies, we've really leaned on the cancer field a lot. So cancer cells for quite some time have been thought to have metabolic changes underlying a lot of their either change in cell state or in their proliferation, like metastasis. And so they've developed a number of drugs to inhibit many of these metabolic enzymes that we're studying. And so we're also looking at co-opting some of that work in the cancer literature and moving that into the heart as well.
It's interesting. A lot of therapies that are now being thought of for cardiovascular disease have their origins in cancer, not just from imaging modalities, but to new therapies to target things like cell death in cells that you don't want around that much in the heart. From some of your work, you've shown that there's these types of fibroblasts, which are also parts of cell component of your heart in conjunction with things like cardiomyocytes that provide the contractile force. You've shown that metabolites can actually affect how these fibroblasts kind of differentiate and change into a different type of cell. I wonder if you could describe that a little bit in terms of how that affects outcomes from an injury to the heart.
Yeah, so I guess one of the ways we think about the mitochondria, and we think specifically about metabolite signaling, is any work or any particular job the cell is trying to do. So if you think about a cardiomyocyte, for example, contraction, or a fibroblast producing this extracellular matrix or these collagens, for example, that has to be linked tightly to energy production so the cell can carry that out. And in tune, energy production is also linked to that function. And so one of the things we've been trying to understand is how these metabolites, or little substrates that are required for producing the energy, actually signal to the nucleus for changes then in gene expression to actually carry out either a change in cell fate or a change in function. So in the context of what I was talking about today, it's how a fibroblast becomes this myofibroblast, this very synthetic, large contractile cell, which we think is definitely a contributing component to fibrosis and heart failure progression. So understanding how some of those metabolites signal and how we can perhaps target some of those metabolic pathways is something we're very interested in.
Taking the analogy to cancer a little further, one of the problems with cancer is that you get these changes, but then you also get uncontrolled cell growth. One of the differences in this is that the myofibroblast might actually be able to go back to more of a resting state, and you might not necessarily have to kill it, right, to be beneficial therapeutically. Are there ways that you're thinking of trying to de-differentiate a fibromyoblast back to a state where it may be more resting?
Yeah, I think that's like the ultimate take home is can you do that without actually killing the cells? There are therapies aimed at that. Mm-hmm. There's work at Penn where they're trying to, using CAR T therapy, for example, to actually directly kill and destroy the fibroblasts and having, they think, beneficial outcomes.
Again, taking from the cancer field,
yes,
right?
Yep, exactly.
Where they can create a T cell that's a cell that's designed to actually go and kill cells that either look foreign or been infected with viruses, but they're using those to specifically target cancer cells and kill those. So a mechanism, again, taking from the oncology field and trying to apply it to cardiovascular disease.
I guess our thoughts on that is that reverting those cells may be a more beneficial way to go. Mm-hmm. The fibroblasts are important in the homeostatic heart for maintaining that extracellular matrix in that ever-changing environment. So the heart's constantly contracting. And so we think just eliminating those cells might not be the best approach. And so the goal that we're trying to do is basically block the signals that maintain that disease phenotype. In other words, can we start to get that cell to, like you said, de-differentiate or shift back into a more homeostatic or baseline state?
One of the things that really struck me in your presentation today was the amount of stuff the heart produces and it sends out, right, in the vasculature and maybe affecting other organs. And you mentioned a couple metabolites like glutamine. What do you think the net effect is of a heart that's in heart failure producing these things on other organs in the body? And how might that actually be one of the reasons why heart failure is such a difficult disease to treat?
Yeah, I think you hit the nail on the head. There's many labs that are really trying to understand the various contributions of different organ systems to the failing heart. And a lot of interest right now, as you likely know, is in HFpEF, heart failure with preserved ejection fraction. Right. And these patients have numerous underlying etiologies, diabetes, obesity, hypertension, all of these things that really have multiple organelle components, skeletal muscle, exercise intolerance is one of the key features of that disease. And so I think understanding the signaling, not just between cells in one organ, but between multiple organs is really where the fields are starting to shift and move to. We're definitely early on in that game, but it's something that we're very interested in.
beauty of science is that we're often very early on, and it keeps us all interested and curious about the next steps, but it also leads to very excellent discoveries in therapeutics that can be used to treat patients. Thank you very much for your time. Thank you for coming to Houston. Thank you for a great talk today and the seminal work that you do in the area. I think one of the greatest compliments I heard today was when Dr. Tackmeyer, right, the godfather of metabolism,
Yes.
said today's presentation was one of the most extensive metabolism seminars he's seen perhaps in his lifetime.
Well, that's definitely not the case. I think he was being overly generous with that compliment. But yeah, he is the godfather of cardiac metabolism that all of our work touches on or leans on in some way. So it was great to
that
have.
has to be a personal milestone, I imagine.
It was great to have him there and be able to talk with him today.
Well, thank you again for coming.
Thank you. Really
Great
appreciate
pleasure
your
meeting you.
time.
Yeah.